Welcome to Ask and Learn.
Hello students, here we are being asked questions regarding the activity of hibernating M. nattereri, which were recorded at the Greywell Tunnel, United Kingdom over a 24-hour day, January 2008, 2009 and 2010 combined. The first question asked is, one hypothesis proposes that foraging occurs during hibernation season and that M. nattereri show the highest daily activity levels when risks from predators are reduced. Now the question asked is explain how natural selection most likely resulted in a common daily cycle of activity among most individuals in the population. So the natural selection most likely resulted in a common daily because individuals that were active during periods of reduced predator risk had a higher chance of survival and reproduction. So over time, the genes associated with their daily cycle of activity would become more prevalent in the population, which leads to a majority of individuals displaying this behavior. Another question asked is this propose a hypothesis regarding the effect of light on the activity of hibernating. M. nattereri described the negative control treatment in the experiment that could be performed to test this hypothesis. So a hypothesis regarding the effects of light on the activity of hibernating M. nattereri would be that the bats are more active during periods of darkness, as this would coincide with the reduced predator risk and the negative control treatment in an experiment to test this hypothesis could involve monitoring the activity of the bats in a constant light environment. And this control treatment would increase the validity of the results by allowing researchers to compare the activity levels of the bats in both light and dark conditions and helping them to determine if light has a significant effect on their activity or not. The researchers decision to average the number of events detected by the motion sensor per 5-minute block every January 2018, 2008, 2009, and 2010 was justified because it helps count for any potential variation in activity levels between years and provided a more accurate representation of the overall activity patterns of the bats during hibernation. Another question asked is identify a 1 hour window during the 24-hour day that M. nattereri is most likely to be active foraging. So the M. nattereri is most likely to be actively foraging during the 1 hour window is between 6 and 7 hours, and this is supported by the fact that the mean activity of hibernating M. nattereri is highest during this time period, indicating that the bats display nocturnal activity. Another question asked is a student claims that the genetically controlled circadian rhythm in M. nattereri is less than 24 hours, but that exposure to a daily cycle of light and dark and trains the bats to 24-hour cycle of activity. Test this claim as student group of hibernating M. nattereri was moved to an artificial environment under constant darkness where their activity was monitored using infrared motion detectors over 31 days in the month of January. Now the question asked is predict the observed activity pattern that would support the student's claim. So to support the student's claim that the genetically controlled circadian rhythm in M. nattereri is less than 24 hours, but that exposure to a daily cycle of light and darkness trains the bats to a 24-hour cycle of activity, and the observed activity pattern in the constant darkness environment should show a gradual shift in the timing of peak activity over the 31 days. This would indicate that the bat's internal circadian rhythm is not exactly 24 hours, but when exposed to a daily cycle of light and dark, that their activity became synchronized to 24-hour cycle. I hope you found the video useful. Thank you.